not a problem. Looking forward to it.
Me too. Quality and scientific excellence. This is a hot topic in the bioscience boardroom. And so I was hoping we might be able to jump in with a little bit of, perhaps you can describe your background to us.
Okay, so so my background, my PhD is in molecular pathology. I've spent most of my career working in drug development, but primarily I've been focused on developing novel models for applications in drug development research, development of novel animal models. So I spent a lot of time working on the commercialization of what we call humanized animal models. These are animals that have been modified to express human genes, or animals that have been modified to be able to engraft human hematopoietic stem cells so that they then go on to express cells of the human immune system. Also worked a lot in oncology development of patient-derived primary in vivo models. I've done most of my career, certainly the last 60 years or so, as being in the CRO industry. So engaged in working with big pharma or biotech companies, executing their studies on their behalf, either because they did not have the scientific understanding of the models and therefore they were outsourcing, or sometimes it's just a capacity issue. And as a result of not having capacity within their own facilities, they outsource. Spent several years as a study director, and then my career has moved on from there: study director, senior study director, director of scientific operations, and then where I am now as vice president, global quality and scientific excellence. So for a global organization, all our quality divisions report into me. And then I also have a role in ensuring that our clients' expectations of the quality of the science that we execute on their behalf is met, as well as ensuring that any internal studies that we execute to generate data to support our marketing efforts are executed in the highest standards and integrity.
And all of that is super important when you're talking about the drug development pathway. Each one of those areas that you've mentioned are key to success, right? I mean, if you don't have the right capacity, if you don't have all of those standards in place, understanding of the basic models, how do you go about choosing some of the models when you've got so many different possibilities in front of you?
I think the key is understanding the questions that are being asked. Once you have an understanding of the question, you can review the models. Models have their limitations, and often we talk about models in terms of how they can be applied, but I often find that it's more important to understand the limitations of a model. When you understand its limitations, it helps you select the most appropriate model to answer the questions that you're looking to address. So yeah, that's where I think the most important focus needs to be, is understanding model limitations. No model is perfect.
Can you give us an example, Leon, what you're talking about when you say model limitations?
I think one example would be as it relates to humanized models. You know, there's a lot of interest in the industry in humanized models, especially because of the advances being made in the field of immuno-oncology. And a lot of the biologics that are being developed are specific to human targets so they don't cross-react with mice or with rats. So, we've developed humanized models of the immune system. So these are mice that engraft either hematopoietic stem cells or peripheral blood mononuclear cells, and those animals can express functional aspects of the human immune system. But if a client is targeting NK cells, for example, you've got to understand that not all of the humanized models express NK cells, or some of them express NK cells, but those NK cells are not functional. So again, you've got to understand the limitations of each of the models, and that will help you select the appropriate model. So in this case, it would be a model that expresses NK cells, and those NK cells are functional.
Yeah.
If a client is interested in combinatorial treatments, treatments where you're looking to do a first round treatment targeting T cells, and then your second round treatment is looking at NK cells. Well, many of the models that express human functional NK cells do not express functional human T cells. So again, it's a limitation. You have to understand that limitation.
Thank you. That was very, very insightful and well said. I don't think I ever realized that some of these models, the humanized ones, what they're expressing isn't necessarily functional. I guess I had always assumed that they would be.
No, I mean, again, I think what you've got to do is you've got to understand each of the different methods of humanizing the animals, and often to address a question, you may have to end up utilizing two different models to address the different aspects of the questions that are being asked, and executing two different studies as opposed to trying to address all variables within a single study.
Okay, so how do you deal with some of that and adding into, going more into a quality regulatory type role? Is it about making sure that the systems and the measurements and the documentation is there?
Well, for my quality role, because I've been involved directly in the execution of research for so long, as well as having run a laboratory operation, I have both an understanding of the needs of the client in terms of, speed is very important. The time from, signing of that quote to initiation of a study and delivery of the data. But that has to be balanced against the need for quality. Quality includes things like ensuring that the documentation is solid, so that if the study needs to be repeated, or that all of the details, all of the key details that need to be captured have been captured. Not only that, but just in the execution of these models, the models are quite sensitive. So you need to be able to execute them in an identical manner each time that they're run, so that you know that the data that you're going to be achieving at the end is scientifically valid and reproducible, and that the clients are able to make the appropriate decisions based on the data that's being received. So what goes into that? It's optimizing models and writing the standard operating procedures so that the models can be executed in the same manner each time. It's training so that the staff can execute the procedures again in the same manner each time. It's putting all of the necessary pieces in place to ensure that you're operating a finely tuned machine.
That's so important. You talk about repeatability, and quality really has a key player in there. You talk about SOPs, and so important, right? The QCTMs, the SOPs, and all of these things in order to make sure that each time we want to make sure that the validity of your measurement is precise, that you're truly getting to the answer of the question that you want. And part of that, I would say as well, from a quality perspective, you've got to be able to make sure that your initial question is well drafted,